Hey everyone, it's Mr. Stratton. Today I want to talk to you about lipids. If you look at the blood samples to the side there, you'll notice that they're quite different. The one with the red cap is actually a normal blood sample, with plasma on the top and red blood cells on the bottom. But the one with a purple cap actually has what's known as hyperlipidemia, which is too much lipid in the blood. So that top yellow portion is actually fat. So this person could just have a very poor diet and be in very poor health, or it could also be that they just didn't fast before they went for their blood sample. But in either case, having that much lipid in your blood is not good for you. So what we're going to focus on, our essential question for this is, how is the structure related to the function in living organisms? Now the functions we're worried about, primarily long-term energy storage, the structure of cell membranes, and the hormones. Now, lipids are not truly polymers. They aren't made up of just one repeating monomer. Instead, they're macromolecules, but they're made up of a couple of different small pieces. So the ones we're worried about primarily are glycerol and fatty acids. And then the notable molecules we're going to be talking about, triglycerides, which is a fancy name for fat, phospholipids, and cholesterol. So triglycerides, the first one, their structure is a glycerol backbone, which is a sugar alcohol, with 3 fatty acid tails. So tri in triglyceride tells you that it's got 3 tails. So those fatty acid tails are just long chains of carbon and hydrogen. So the way that you put these together is through a dehydration synthesis reaction, right? The hydroxyl group, which is actually part of a carboxyl group on the fatty acid tail, combines with a hydrogen from the hydroxyl group on the glycerol and forms what's called an ester linkage. So it's also broken down, just like other biomolecules, through hydrolysis. Now, the important thing about this is that that long chain of carbon and hydrogen in the fatty acid tail makes this extremely nonpolar. Carbon and hydrogen have very similar electronegativities and share electrons equally. Thus, there's no polarity in those tails at all. This molecule is very hydrophobic as a result of that. It does not like water. The tails can actually occur in a couple of different structures. The two we need to really worry about, saturated tails have all single bonds. Saturated means full. So all the spots to bond are full. Whereas unsaturated means that there's at least one double bond there. And that double bond, if there's more than one, it's called polyunsaturated. But that double bond causes a bend or a kink in that tail. And that's going to be important for the actual structure of the molecule and the physical properties of that. Saturated fats are typically animal fats, and they are solid at room temperature. So like lard, manteca, or butter, mantequilla, what you're looking at there, those are saturated fats. Think of it this way. A molecule is going to be either a solid, liquid, or gas, depending on how close its molecules are together. Now, if you've ever gone camping with your family, you would know that if you're in a tent and you're all in sleeping bags, the easiest way to get the most people into that tent is if you're all in a single file line, just like these saturated tails, straight. Now, on the other hand, unsaturated would cause there to be a bend in that molecule, and you would not, if you're sleeping and you're bent in your sleeping bag and you can't touch other people, then you're not going to fit as many people into that tent. And so the result is that unsaturated fats, which are primarily plant fats, are oils at room temperature. Now, there is an exception to this. There's something called a trans fat, and that's when the hydrogens are across from each other. Trans means across, like, for example, transatlantic flight. So trans fats have the hydrogens across from each other at the double bond, and they're actually straight, and they're solid at room temperature. Now, in nature, there's not really that many. Primarily, this is caused by what's called artificial hydrogenation, where we humans put extra hydrogens in there to try to straighten out these molecules, to try to make an unsaturated oil into a saturated fat. Something like Crisco, which is an artificially hydrogenated, it's actually hydrogenated soybean oil. All right, the function of triglycerides is long-term energy storage. So we have the ability to store energy using fat in those long chains, those long fatty acid tails, with all those carbons in there. There is a lot of energy. In fact, there's more than double the energy, the number of calories in a gram of fat versus a gram of carbohydrate. And we actually store our triglycerides as fat droplets within cells, especially within adipose cells. Okay. A different type of lipid we're going to talk about is phospholipids. So phospholipids are very similar to triglycerides, except they have, instead of 3 tails, they have 2. So there's a glycerol backbone, 2 fatty acid tails, which can either be saturated or unsaturated, and then in place of that 3rd tail is a phosphate-containing group. Now, you don't have to worry about the structure of that phosphate-containing group. In freshman biology, you really just drew this as a circle with 2 tails on it. So the structure of this leads to an interesting feature, which is the fact that it's amphipathic, which means dual tendency. So the head, which has that phosphate in there, is actually polar, and it likes water. It's hydrophilic. The tails, on the other hand, carbon and hydrogen, very nonpolar. So this molecule, it's torn. It's got a head that likes water and tails that don't like water. So structure-wise, it looks a little bit more like this. There's a bunch of different representations, but you remember this from the cell membrane. Now, in the cell membrane, it forms a bilayer, which has heads facing out, tails facing in, and that's all because the environment around the cell and within the cell, that's aqueous. It's water-based. And so the phosphates facing outward like water, the tails hang out together in the middle because they hate water, and the result is this bilayer. But think of some other ways that you could arrange phospholipids that would also allow the heads to face towards water and the tails to face towards, well, other tails. There's a couple other ways you can come up with. The final lipid we're going to talk about is completely different. This is a sterol, and it's got these 4 interlocking rings. This is the base skeleton. Add a bunch of different things on there, and you get a bunch of different molecules. So you don't have to memorize the structure. Just if you look at this, you can recognize and say, oh, that's a sterol. Now, sterol is part of the name of the main one you would know, which is cholesterol. So found in the cell membrane, it actually helps our membranes to stay fluid at low temperature. So it's actually fit in between the fatty acid tails within the membrane, and the result is that the tails can't get as close together, and, well, your membrane doesn't freeze as easily. There are some people that naturally have more cholesterol in their membrane, particularly people like the Inuit, and the result is they're much less susceptible to frostbite than you or I probably would be. So it's a precursor as well to produce sex hormones and vitamin D. So if you look at that chart to the side there, you start with cholesterol at the top, and you can produce a whole bunch of different hormones you've probably heard of. For example, progesterone, cortisol, which is a stress hormone, estrogens, which are those ones in the pink triangle, testosterone, all of that can be produced from cholesterol. And so you need cholesterol in your diet to be able to make those hormones. So the way those hormones work is they're chemical messengers. They flow through your blood, and they can actually cross the cell membrane, enter the cell, where they can bind to a receptor and then bind to the DNA and change gene expression. So if you were to take some extra testosterone and inject that today, what would happen is it would go in, it would alter your gene expression, it would cause you to produce more hair, it would cause certain parts of your body to grow, including making your voice deepen, and it can also cause acne. So that would lead to some changes in your body, all because the gene expression would change. And that's what happened when you went through puberty. What I want you guys to do now for your reflective practice is structure and function for triglycerides. How are they related? And then phospholipids, how are they different in terms of their structure and function? And finally, in the last one, I want you to predict when an organism would want more unsaturated fatty acid tails. So there's actually something called homeoviscous adaptation, where an organism can change the number of saturations in its fatty acid tails in its membrane. So what would be the benefit of that? And then go ahead and justify your prediction using the properties of phospholipids. Okay. Thanks for watching.